Good morning. It's April 23rd, and this is your daily brief in science. Here's everything you need to know. A surge of controversy surrounds a COVID-19 vaccine effectiveness study that the CDC's Morbidity and Mortality Weekly Report blocked from publication, with officials saying methodological concerns stalled the release. The Washington Post first reported the delay, noting the manuscript previously claimed vaccines reduced emergency visits and hospitalizations among healthy adults by roughly half in the prior winter. The decision to block publication came from the U.S. Department of Health and Human Services, with spokesperson Andrew Nixon citing multi-level review standards and confirming the manuscript was not accepted for release. The report also references Daily Kos articles to illustrate claims about Kennedy's actions and their consequences, alongside notes on Daily Kos's funding model. Kennedy has publicly criticized certain vaccine policies and pushed for transparency and medical choice, while denying he is anti-vaccine. His role is framed as shaping vaccine messaging in politics and administration. Kennedy's history of anti-vaccine rhetoric and calls for changes in guidance have drawn scrutiny from lawmakers and experts, though he maintains his goal is transparency and medical choice. The incident underscores fierce debates over transparency, scientific independence, and how real-time vaccine effectiveness data should be reported amid evolving public health and political pressures. The coverage highlights the tension between political oversight and scientific publication in public health communications, with the Associated Press placing the story in that larger context. The piece notes that the AP's reporting comes as debates about transparency and political influence in public health continue. Experts familiar with the VISION network say the study design is well established and widely used in high-profile journals, suggesting the rejection may reflect political interference rather than methodological flaws. Critics, including former CDC official Dimitra Daskalakis, describe the move as suppression or cherry-picking influenced by leadership bias, arguing it undermines transparent scientific expertise. Context includes internal CDC dynamics, resignations, and disagreements within vaccine policy advisement, framing the current dispute. Astrocytes, the brain's support cells, are now shown to form extensive, long-range networks in the mouse brain that connect distant regions through gap junctions. These networks are dynamic and respond to sensory experience. After whisker trimming, the pathway from the sensory region contracts and rehangs with different astrocyte partners, indicating experience-dependent remodeling. Researchers used a custom tracing approach with virus-delivered network tracers and brain clearing to map active astrocyte networks, revealing pathways that can link areas not connected by neurons. In vitro validation confirms the fusion protein localizes to gap junctions, and that biotinylation occurs within the gap junction vestibule, with low native biotin in the central nervous system minimizing background and aligning labeling with enzyme reach. The study, led by New York University Langone Health, was published in Nature on April 22, 2026. And funding came from National Institutes of Health grants and various foundations, with disclosures noting researchers' affiliations and potential conflicts of interest. In vivo experiments showed that, after regional tracer injection, biotin administration, and light sheet imaging revealed infected astrocytes and neighboring in-network cells with biotinylated cargo, confirming gap junction-mediated spread. Double knockout of astrocyte gap junctions, CX43 and CX30, markedly reduces network size, indicating dependence on astrocyte gap junctions rather than vasculature. While networks exist in humans, it remains unknown whether the same regions are connected as in mice, and experts unaffiliated with the study view this as a fundamental advance in nervous system structure, though the functional relevance remains to be clarified. Quantitative mapping aligned with the Allen Brain Atlas showed largely distinct motor cortex networks, with more overlap in frontal and hypothalamic regions, and some contralateral enrichment suggesting a complex topology across the brain. A new fluorescent tool lights up only when it binds its target, cutting background noise and delivering ultra-clear, multicolor images in live cells and animals. Researchers from Albert Einstein College of Medicine and the Salk Institute report engineered VIS-Fb nanobodies that fluoresce solely upon binding, enabling high-precision visualization of proteins in real time. The study, published in Nature Methods on April 22, 2026, demonstrates broad utility across mammalian cells and in vivo models, with NIH and foundations funding the work. VIS-Fbs achieve precise imaging of central nervous system activity in neurons and astrocytes in behaving mice and track developmental and drug response dynamics in zebrafish embryos. The platform has been validated across multiple cell types and species, allowing calcium signaling imaging and real-time developmental readouts with reduced background. The VIS-Fb system supports over 20 fluorescent proteins and biosensors, is modular, and can swap nanobody modules or fluorophores to tailor targets and readouts. Some variants even offer optogenetic control, expanding how researchers can manipulate and monitor biological processes. In mouse models, high-contrast neuronal activity imaging is achievable during behavior, while zebrafish models reveal rapid developmental changes in drug responses. The collaboration lists Axel Nimmerjohn and Vladislav Verkusha as co-corresponding authors, with authors from Salk, Einstein, and partner institutions. Funding came from NIH and multiple foundations, emphasizing the platform's potential for studying cell signaling, development, and disease progression with greater precision. The Nature Methods report highlights VIS-Fbs as a flexible tool for diverse biological questions, spanning cancer, neurodegenerative and infectious diseases, developmental biology, and neurobiology. Coastal cities face increasing flood frequency and intensity as climate change advances, making proactive adaptation essential for resilience and equity. A new analysis maps storms from 2012 to 2017 to identify at-risk populations and links risk to poverty, race, education, and age. The study emphasizes that risk comes from floodwater, land subsidence, impermeable surfaces, and social vulnerability, creating a complex risk picture. Vulnerable groups highlighted include people living in poverty, minorities, those without a high school diploma, young children, and the elderly. The authors advocate for mitigation measures such as protective structures, preserving wetlands and estuaries, and using permeable surfaces, while noting challenges from high property values and relocation. Officials and emergency managers are urged to use the analysis for planning, response, infrastructure protection, and public communications to inform communities. The research also stresses that outcomes depend on storm paths, rainfall patterns, and future climate changes, underscoring the need to prioritize vulnerable populations in planning. Scholars call for local planning that reduces harm to socially vulnerable groups in future floods, linking the scale of at-risk populations to where people live and how cities are built. The piece highlights the importance of ongoing science communication and support for science journalism. It concludes that flood disasters hinge on location and urban design, reinforcing the case for proactive planning to protect vulnerable communities. A new gene therapy approach for OTOF-related deafness shows durable hearing gains that could reshape treatment options. In a multi-center trial spanning 8 centers, participants experienced ABR thresholds falling from about 97 decibels at baseline to roughly 42 decibels after 2.5 years, with behavioral tests confirming meaningful improvements across the group. The study, published in Nature, describes sustained hearing restoration and positions gene therapy as potentially one-and-done for many patients, though longer follow-up is needed to confirm long-term safety and efficacy. Early diagnosis and genetic screening, plus biomarkers, may help optimize who benefits most in future applications. No dose-limiting toxicities were observed within 6 weeks after injection, and adverse events were generally mild, with only a few grade 3 events like decreased neutrophil counts that were clinically manageable. Adults showed less robust gains than children and teens, but several adults still experienced meaningful improvements, with no serious adverse effects beyond mild immune fluctuations and rare inner ear inflammation. The therapy uses AAV1 vectors delivering a codon-optimized human OTOF transgene aimed at cochlear hair cells, illustrating precise molecular engineering for a sensory disorder. Regeneron's work could represent the first FDA-approved gene therapy for deafness, with U.S. trials likely required. The Chinese trial will continue through its regulatory process. Next steps include exploring multiple dosing and longer follow-up to assess durability, and comparing outcomes with cochlear implants, noting that benefits in music perception and noisy environments may exist, but implants may remain the default for many patients. The report reflects comprehensive, industry-focused coverage from STAT+ and underscores robust longitudinal data supporting safety and efficacy across a broad age range, highlighting the study's multi-institutional collaboration across 8 centers. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.